Now it's my pleasure to introduce our first speaker, Professor Simon Billinge. Professor Billinge has more than 25 years of experience developing and applying techniques to local structure in materials using X-ray, neutron, and electron diffraction, including the development of novel data analysis methods, including graph theoretic, artificial intelligence, and machine learning approaches. He earned his PhD in material science and engineering at the University of Pennsylvania in 1992. After 13 years as a faculty member at Michigan State University, in 2008, he took up his current position as professor of material science, applied physics, and applied mathematics at Columbia University. He's also a physicist at Brookhaven National Laboratory. It's my pleasure to turn the talk over to Professor Billinge.
Okay, thanks, Robert.
We're still
have
turning you over.
Okay.
You should have the screen here pretty soon.
Okay, so now maybe you can see me.
Yep.
Now let's see if we can see my presentation. And let me get spotlight. Okay. Okay, how do I get that screen back again?
So we have your intro, your title slide. It looks great.
Okay, that's good. Somehow or other, I've got it covered up on mine by my picture of myself. Okay, now I can see it. All right, good. So thank you everyone for coming. It's really a pleasure to have this opportunity to talk to you. And what I want to present today is actually developments that we've been making, and this is a collaboration with Partha and the team at Nanomegas. It's also a collaboration with some other people, such as Mitra Taheri, who's at Johns Hopkins University. And the basic goal is to actually develop the capability to do scanning nanostructure electron microscopy. So we want to do scanning electron microscopy, but where the contrast feature that we're interested in is actually nanostructure. So I'll tell you about this, and I'll spend some time introducing pair distribution function method, which is actually the signal that we're going to image. Okay, how do I advance the slides? Sorry about this, I don't quite know how to get control back. I guess I can't. Okay. So a little bit about myself. Robert gave an introduction, but I'm a professor at Columbia University. So this here is a picture of Manhattan, and this green part here is Columbia University. And my joint appointment at Brookhaven allows me to use actually the very intense X-ray scattering sources they have there, and also the microscopy facilities at Brookhaven. But really, I'm a methods person. I like to develop new methods for studying nanostructure. So if we take a very naive view of electron microscopy, it's basically you've got a heterogeneous sample, and you want to actually see an image of it. And the traditional approaches are that you might do a bright field image, and that's a bright field image here of a bacterium. And there's lots of dark areas and light areas, and there are some black dots. And this is really measuring electron density as well as I understand it. And some of these black dots are because actually the sample makers that introduced this is not my work. It's the reference is down here, but the sample makers or the researchers that introduced gold nanoparticles into this material. And so some of these black dots are gold nanoparticles. They're regions of high electron density, and the question is which. And of course, you can rather straightforwardly, if you have an image in a diffraction pattern, if there's a particular point in a diffraction pattern which is coming just from those nanoparticles, you can do a dark field image. So what you do is you look at your diffraction pattern, you find a signal from the gold nanoparticles, you put a selected area aperture over that portion of the diffraction pattern, and then you re-image, and then you see these very light spots over here are actually coming from the gold nanoparticle. So that's called dark field imaging, and it's of course very familiar to everyone. The point here, though, is that you can get images and you can use different contrast mechanisms, so different signals that you use to obtain contrast. But let's say, of course, you wanted to make a map of the elemental composition where the carbon atoms are and where nitrogen atoms are and where oxygen atoms are. What you can do is you can do an EELS experiment, and this is an EELS spectrum here, which is the energy loss of the electrons after they go through the sample, and then you can see features in this spectrum. And you can do the same thing. You can say, well, let me select some feature in this EELS spectrum, and then I'll make a two-dimensional image of that thing. And again, I'm sure that you're all extremely familiar with this, but whatever the signal is that you're using to make as contrast, you're actually getting an image about that thing. So in this case, because we're looking at an absorption in the EELS spectrum, we're seeing where the element that produced that signal in the EELS spectrum is located. And this is how we do sub-nanometer resolution elemental mapping. So bottom line is you can do microscopy by looking at a function of position, but you can use any kind of signal. And in this case, it was counts in a region of my EELS spectrum where I expect to see a strong emission from copper, let's say. And this throws open the door to coming up with all kinds of different microscopies where anything that's any quantity that's measurable from your sample, in principle, you can find that as a scalar signal. And in my group, we call them the quantity of interest. And then we can map that as a function of position. So we have a lot of fun in the group actually coming up with different kind of quantities of interest. So a quantity of interest is just a scalar quantity that you can compute from the signal coming from your sample. So then we can ask the question, what if we want to get maps of nanostructure? So I give a particular example, which is just pulled out of the air. It's not a real one, but let's say that you have a material and it has Fe3O4 particles in it, and it has Fe2O3 particles, and you want to see where the Fe3O4 is versus the Fe2O3. And this kind of microscopy would be rather difficult if you were trying to use just bright field imaging. They have a similar contrast and they both have iron and oxygen, so it would be difficult to image this using elemental mapping. But these two structures have, these two phases have very different structures. Okay, now if these are actually powders and you could find a distinct signal in the diffraction pattern, you could use dark field imaging, which I already showed you for the bacteria with the gold nanoparticles in. But if these are nano-crystalline, so they're very small particles, or if it's amorphous material, the signal in reciprocal space in a diffraction pattern is very broad, and the signals from these things will overlap, and it will become very difficult to actually make a spatial map of those structures using dark field imaging. So the question is, can we do better when we want to get maps of nanostructure? Another point I want to make is maybe you don't want to just do phase mapping. Maybe you want to ask a much more subtle question. For example, let's say that we have nanoparticles of Fe3O4, and some of them have some tin in them, so they're doped with tin. And actually what you want to know is the distribution in space of the tin-doped samples. Some of them have tin in, some don't have tin in. You want to know where the tin-doped nanoparticles are, for example. It's just a crazy example I came up with, not a real one. But then you would need a much more subtle signal you can't just do a dark field image in a kind of traditional sense. What you would want to do is you would want to do some kind of structural analysis of that thing and extract a signal that was sensitive to the doping. So it might be the lattice parameter of that material. And then the quantity of interest would be the lattice parameter of that material. And you could then plot the position of the nanoparticles as a function of the lattice parameter, and you would see some gradient in the tin content as a function of position. So the point being that if you can actually extract a structure from your sample and you can do modeling on that structure, you can get all kinds of different contrast mechanisms to answer many, many different scientific questions. So what I'm going to tell you about now is actually Electron PDF. Maybe you do, or maybe you don't know what PDF is, but I'll explain it to you. But PDF is the technique that we use for getting structural information from nanomaterials. So what is PDF? So PDF stands for the atomic pair distribution function analysis. It's a pair distribution function analysis of powder diffraction data. And let's close that. Okay, so of powder diffraction data. And it's particularly useful when you have nanomaterials or amorphous materials. So it's the best approach for getting structural information from samples that have very small nanoparticles. It basically, the basic approach is that it takes diffraction data from reciprocal space and it does a Fourier transform to represent it in real space. And the reason that's useful when you have nanostructured materials is that for nanostructured materials, the features are very spread out in reciprocal space. And through the Fourier transform, we make them very sharp in real space. So it's just a diffraction technique. It's kind of like powder diffraction, but it's particularly good for studying nanoparticles. So the approach is the following. You actually have a powder, or you have a distribution of your nanomaterials, or you have an amorphous material, and a beam comes in and it gets scattered at some angle, and you get a diffraction pattern. And because it's a powder, you get a one-dimensional diffraction pattern. If you have a 2D detector, you get a two-dimensional diffraction pattern that you integrate around the rings to get the one-dimensional diffraction pattern. And then the diffraction pattern can look like this up here. So we do corrections to the data to make it into an absolute structure function, and then we do a Fourier transform. So this is just a sine Fourier transform here, and that results in the PDF. So the very beautiful thing about the PDF function is it's a very intuitive function. So on this axis here, this axis is a probability, and on this r-axis, this is a distance. And so the information in representation is that the function here is the probability of finding two atoms separated by this distance. So my PDF here of this material has a very sharp peak at this position. This is 2.75 angstroms. It means that in this particular material, there's a very high probability of finding two atoms separated by 2.75 angstroms. And then all of these other peaks are coming from other interatomic distances in the material. And we can kind of reconstruct the three-dimensional arrangement of atoms that's consistent with this set of interatomic distances. So to get a kind of intuitive grasp of how the thing works, this is actually some real data. It's X-ray data coming from C60 carbon nanotubes. And in the top right panel here, we have actually the diffraction data, the one-dimensional diffraction pattern. And down here in the lower panel on the right, we have the Fourier transform of that, which is the PDF. And C60 is made of carbon, so it has a carbon-carbon bond at 1.4 angstroms, so that strong peak is at 1.4. Then the second neighbor distance is this one here. Third neighbor distance is across the hexagon on the buckyball. That's this distance and then this distance. And the buckyball is actually made up of hexagons. So this is a hexagon and pentagons. These 5 atoms here are in a pentagon. And if you look carefully at the pentagon, it has a near neighbor distance and it has a second neighbor distance, but it doesn't have the third neighbor distance directly across here. And so the pentagon is contributing to the first peak and to the second peak, but not to the third peak. And the hexagons are contributing to the first peak, the second peak, and the third peak. So we can immediately, without doing any modeling, get the ring statistics, the relative amount of 5-fold rings and 6-fold rings, just by determining the relative intensity of the third peak and the second peak. But if we know the structure of C60, what we can do is we can actually reconstruct this whole signal here in a self-consistent way, and we can understand exactly the full structure. And one of the amazing things is if you sit on this carbon atom here at the bottom of the ball, and you look at the carbon atom that's the furthest distance away, it's across the diameter of the ball here, and the ball is 7.1 angstroms, and the last sharp peak in the PDF is at 7.1 angstroms here. So it's, in my view, it's kind of almost like magic that I have a structure here with well-defined interatomic distances on a ball that's only 7 angstroms in diameter. And I get these sharp peaks in the PDF which go up to 7, and then they don't go beyond that. And I simply get it by measuring the powder pattern and Fourier transforming it here. So this signal here gives this one. And if you look out here in the higher R region, what you see is actually there is some structure here. It's just rather broad, diffuse peaks. And the first peak is at 10 angstroms, and it turns out that 10 angstroms is the ball-ball separation. So in this low R region here, we're seeing the arrangement of atoms on a single ball. And in this high R region here, we're seeing the arrangement of the balls themselves. There's a second peak at 14 and a third peak at 17, and that's root 2 times 10 because this is an FCC structure. And the third neighbor peak is root 3 times 10, which is 17. And so actually these peaks out here are telling us how the balls pack, and these peaks in here are telling us how the atoms pack on the ball. And we're getting all this information for free simply by doing a powder measurement, doing the corrections, and then Fourier transforming. So we do this routinely with neutrons, we do it with X-rays. So we can measure a PDF. We can then calculate a structure from a PDF. So if we use this, again, we'll stick with carbon, but we'll go to graphene now. And to calculate the structure, to calculate the PDF of a structure, you put an atom at the origin, and then you go out some distance until you find another atom. So the first atom we find away from this guy is at 1.42 angstroms. That's the carbon-carbon distance. So we put a peak at 1.4. And then we go further until we find another atom. That's at 2.46 angstroms. This is the second neighbor peak, and that's the one that I showed you on the buckyball. And then the third neighbor peak is this 2.84. That's the one that goes across the diameter of the hexagon here. And this is how we build up the structure in the computer, and we can keep adding peaks, going out further and further, adding more peaks. And we now have rather sophisticated software that can calculate the PDF from a given structure, and we can even do regression. So this is like, if you know what Rietveld refinement is, this is doing Rietveld refinement, but it's basically taking a candidate structure model and extracting the structural parameters by fitting that model to the measured PDF. So we can measure PDFs from nanomaterials, and we can also extract structural information from them by modeling. I'll go a little bit quickly through this, but it's actually helpful to understand how the PDF works. This is actually data, X-ray data coming from cadmium selenide quantum dots. So these are nanoparticles of cadmium selenide, Q6 semiconductor, and this is the diffraction pattern of the bulk. Then these three are diffraction patterns from nanoparticles that are 4, 3, and 2 nanometer diameter. And what you see in the diffraction pattern is that sharp peaks in the bulk turn into broad peaks in the nanoparticle. Something different happens in real space. You have sharp peaks in the bulk, but this peak here, it stays sharp when you go to the nanoparticles. What actually happens in the PDF in the nanoparticle is that you lose intensity in the high R region. So you can see that there's a signal here, signal, 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 and then high R, it gets flat. And for this smaller nanoparticle, it starts getting flat a little bit sooner. And for the absolute smallest nanoparticle, the signal gets flat even sooner. So simply by looking with your eye to see where the signal dies out, the signal are these like broader features here, you can tell that this one is a 2 nanometer diameter nanoparticle because that's where the signal becomes flat. In the case of these, one question that we had was the underlying structure wurtzite or zinc blende? These are two different structural candidates for the cadmium selenide. And what differentiates them is actually stacking. They're made up of corner-shared tetrahedra, but they just stack differently. So in wurtzite, it's an AB stacking along the certain crystallographic direction. In zinc blende, it's ABC, ABC. the question that we had, or that our chemistry colleagues had, was, it's the structure of the core wurtzite or zinc blend. And for the bulk, it was very clear that when we fit a zinc blend structure, it did poorly, and when we fit a wurtzite structure, it did well. But for the nanoparticles, it wasn't so clear. So it appeared like the PDF had failed to answer that question. But what we found out was actually these materials have stacking faults in. And so if it's perfect zinc blend, it goes, the stacking is AB, AB, AB. It's perfect, I'm sorry, if it's perfect wurtzite, if it's perfect zinc blend, it's ABC, ABC. And a stacking fault is where it goes ABC, ABC, AB, A. That's a stacking fault. And you can actually go all the way from zinc blend to wurtzite by introducing sufficient number of stacking faults. And what we found was actually the number of stacking fault density was very high, like 50%. So that means you go A and then you go B, and then there's a 50% chance that the next layer will be A or C, which means that the actual structure of these nanoparticles was neither wurtzite nor zinc blend, but it was actually something in between wurtzite and zinc blend. So a kind of amazing thing from the PDF analysis was not only could we tell you something about the structure, but we could tell you something about the faulting in the material and the stacking faults. As I mentioned, we can get the particle size just by looking at the PDF, but we can also fit functional forms for the different shapes of the nanoparticles. So we can get information about whether the nanoparticles are spherical or ellipsoidal. We can see whether there's a particle size distribution or not, and it's all by fitting the shape of the envelope that kills the signal with increasing R. And it turns out that we can even measure strain in these nanoparticles. So this PDF peak here is the nearest neighbor cadmium to selenium peak. And this is the peak in the bulk at the top. And I've drawn a dashed vertical line at the position of the maximum of that peak in the bulk structure. And if you look very carefully, as the nanoparticles get smaller and smaller as we go down this stack, the center of that peak moves further and further to the left. So what it's actually telling us is that there's a compressive strain as a function of nanoparticle size. Now, these nanoparticles are they're not under compressive stress. We're not applying pressure. So the actual strain is coming from some kind of pressure coming from the surface tension is one way to think of it in these materials. And as the particles get smaller, the curvature of the surface gets higher, and so the compressive strain becomes larger in the smaller nanoparticles. And furthermore, the width of these peaks here tell us about the inhomogeneous strain. So the average strain, the homogeneous strain, is coming from the peak position, and the inhomogeneous strain is coming from the peak width. And as the nanoparticles got smaller and smaller, the width of this peak did broaden somewhat, and it's telling us that there's more of a bond length distribution in the smaller particle sizes, which again is not surprising because the cadmium to selenium bond at the center of the nanoparticle is not necessarily going to be the same length as the cadmium selenium bond near the surface of the nanoparticle. So this is just a kind of a very rapid run through a particular kind of chemical system, showing you the kind of information that you can get if you use this PDF approach. Now, there's been a long relationship between PDF and electron diffraction, so I don't want to claim that we were the first to do that. In fact, some of the early electron diffraction work was to study the structure of gases, and that used a PDF approach back in the 1950s, actually. But what we were able to do in 2012 was to show that you can get actually quantitatively reliable PDFs coming out of an electron microscope, and it's not terribly difficult, actually. What's important about that is that the best PDFs that we normally get with X-rays and with neutrons, you have to go to a major facility like an X-ray synchrotron source, whereas you can quite straightforwardly get PDFs from an electron microscope, which might be sitting somewhere in your lab. So this is an example of the proof of principle where we took actually gold nanoparticles, so 100 nanometer diameter gold nanoparticles, and we drop them onto a holy carbon film here. So these nanoparticles are these black dots here on the edge of the holy carbon film. We take a diffraction pattern, which looks like this. We azimuthally integrate the diffraction pattern to get our 1D diffraction pattern. And then we process it to get the f of q, which is our intermediate scattering function. And then we Fourier transform it to get the PDF. And so the blue curve here is the electron PDF from the gold nanoparticles. And the red curve is the model fit of the gold nanoparticle structure to that measured PDF. And so we were able to actually show that you can apply all of these PDF methods that we've been developing for X-rays to the electron case. Now you do have to be careful because of obviously the scattering coming from an electron microscope is not perfectly kinematical. There are dynamical effects, and that actually doesn't change the positions of any of the peaks in the PDF, but it does change some of the relative intensities. But what we found was that if your sample is thin enough, so a few tens to hundreds of nanometer thick, depending on the scattering power of the object you're looking at, and if your scattering is sufficiently incoherent, so you don't have coherent multiple scattering, that actually things work quite well. And furthermore, if you actually use, let me come back to this, if you actually use precession in your, when you take the picture, you can also get a more kinematical signal that will produce a more quantitative PDF. So the bottom line is that the ePDF works rather well. And in order to help people use it, we developed this ePDF suite software. It's a GUI software built on top of the state-of-the-art algorithms that we developed for very fast and accurate data reduction to get the PDF. It supports high throughput of large numbers of data sets, and we're currently working on the thing that will supersede this ePDF suite 2.0. So the bottom line is that actually you can do these experiments using this software. You can get the PDFs, and it includes modeling software as well, so you can model the PDFs. I don't know what, this is actually some input that I got from Partha, and he wanted me to include this, so I'm going to not say too much about this, but if Partha wants to say some words at the end, he can come back to this. But I think that the main message to come away from here is actually, he firstly summarizes here that the dynamical scattering effects do not affect the positions of the peaks in the PDF. They can have an effect on the intensities, but using precession minimizes those effects. And he's also showing some of his work using ePDF on lipoic acid and hexane thiolated gold nanoparticles. And again, he's getting these show measured data here and then a calculated model and a difference curve underneath. And there's really good quantitative agreement between calculated models and the ePDF. So you can get quantitative structural information using this approach. All right, so now let's say that you can do that. That's nice. You've got some nanoparticles. You drop them onto your holy carbon film. But what we're actually able to do now is to do something much more exciting. Is that if we have the ability to measure these PDFs very, very rapidly and to process the data very quickly, what we can do is we can do in situ measurements. So this is an example of an X-ray experiment where we have a PEM fuel cell, proton emission, what is it, PEM? I forgot what PEM stands for, but anyway, it's a standard fuel cell that you can run with methane or with oxygen and hydrogen, and it will produce power. And we were looking at platinum nanoparticles, which are the catalysts in the supported electrode. And we were cycling this PEM fuel cell, and we were looking at the coarsening of the platinum nanoparticles. But the point is that you can actually do these experiments very rapidly, and you can do them as a function of time in principle. That translates over to the electron diffraction context if you have some kind of environmental cell or you want to do temperature dependence or something like that. If you're able to actually do high throughput you can do actually a thin film geometry. So what we also demonstrated as recently as 2015 is thin film PDF, where you actually go in transmission mode through a very thin film that's sitting on a substrate, and we were able to extract a signal from the film. So there are two diffraction patterns here, a red one and a blue one. The red one is just the substrate, and the black one is the substrate with the thin film, and you can see the signals are almost identical. But if you take the difference, you get a signal, and if you do the PDF analysis on it, you can actually get a very nice diffraction pattern which corresponds to actually the thin film. And when you can start doing this in transmission, you can then start doing spatially resolved measurements. So we can think about doing lab on a chip experiments, where what's actually happening here is that we've inkjet printed dots of nanoparticles onto a carbon paper. And then what we do is we take the X-ray beam and we raster it over this sample, and we see the spot that contain the nanoparticles. And then we can do quantitative nanostructure analysis on this. And this thing I'm showing here, it has a spatial resolution of about 500 microns. So with the X-rays, you're limited somewhat by your beam size, and we were limited to 500 microns in this case. We've since then done 100 micron resolution. In principle, you can do 10 micron resolution, but you start to get challenged at that point. What I'm going to show you at the end is that you can take this idea and you can put it into the electron microscope, and you can actually get spatially resolved nanostructure images at the level of 10 nanometers or less. In the X-ray case, we can also do computed tomography. So actually, we don't have to have the sample sectioned. That's not going to be so possible in the context of the electron diffraction. But I just wanted to show you that when you do this kind of nanostructure imaging, you can see multiple different signals coming from the same volume of space, and you can separate those images. So what I'm going to describe, this thing on the right is actually a catalyst body, which is a porous material that contains palladium nanoparticle catalysts. And what we're extracting here is we're extracting a PDF of the palladium nanoparticle. And the palladium nanoparticles at the surface are much larger, and the palladium nanoparticles in the center of the body are much smaller. So we can actually make a map of nanoparticle size as a function of position in this catalyst body. And if you look on the left, this is actually a cross-sectional nanostructure image of this nickel metal hydride rechargeable battery here. And in these nickel metal hydride batteries, you actually have spiral wound geometry. So what you do is you make a kind of like a jelly roll where you have the anode, and then you have the cathode, and then you have the separator and the electrolyte. And what we can do is we can actually look at these 5 different images here, which are all coming from the diffraction pattern, but we've selected a different QOI from the diffraction pattern, so a different region of interest. So the one on the top left is just electron density. So we're just plotting electron density, and you can see that the steel case of the battery is actually the strongest. But what we can do is we can take a signal that's actually coming from the cathode material. So this is actually a map of the cathode material here. We can also take a signal from the anode material. So you can see that the anode is much, much thinner than the cathode. But you can see how it also winds around in this jelly roll structure. And then we can take a signal that's coming from the steel in the material, and you can see the steel shell around the battery. But you can also see this dotted line here. So it turns out that actually the battery structure is the anode is supported on a steel mesh, and this is actually the steel mesh that supports the anode, and we're mapping that. And then this one is actually coming from a polymer signal. And so we're actually looking where the polymer is in the material. And it turns out that there's a lot of polymer on the outside of the sample. And that's this actually polymer covering with the branding on it. But there's also polymer inside the cathode, because actually the glue that holds the particles, the powder particles of the cathode together, is basically a polymer. So we can separately study the polymer and the cathode, even though they reside in the same volume of space. So this is actually very exciting. You can do many, many different things with this.